So 2024 GCE Biology Paper 2, question 1. Figure 1 shows a generalized animal cell. A generalized animal cell. And so we happen to have the labels A, B, C. Question A, they are saying identify the parts labeled A, B, and C. So A is no other than cytoplasm. B, cell membrane. C, nucleus. Yes, A, cytoplasm. B, cell membrane. C, nucleus. Question B, name two organelles present only in plant cells. Name two organelles present only in plant cells. What are the organelles which are only found in plant cells? And of course, we know them, right? Cell wall and vacuole. And if at all we are talking about those plant cells which are found in plants which conduct photosynthesis, we can talk about chloroplasts. So chloroplasts, cell wall, vacuole, etc. Question C, explain the function of the part labeled B. What is the part labeled B? What exactly is the part labeled B? How do I explain the function when I don't know the name of this thing? So B, if you check very well, B is cell membrane. Okay, it is also known as a selective permeable membrane, which means it selects what goes in and what goes out. Okay, it resists the entrance of some materials, but it also allows so the answer is simply to select what goes in and what goes out of the cell. Simple. Question D, describe the effect on the cell in figure 1 if it was placed in hypertonic solution. All right. Describe the effects, or the effect, one effector. Describe the effect on the cell in figure 1 if it was placed in a hypertonic, yes, hypertonic solution. But the question is, what exactly is a hypertonic solution? When somebody says hypertonic solution, what exactly is a hypertonic solution? All right. Okay, kwa bana hypotonic, kwa bana isotonic. Yes, hypotonic, kwa bana isotonic. Yes. So when somebody says hypertonic, what exactly should come into your mind? That's the question. A hypertonic solution is such a solution whereby solutes have a higher concentration than the solvent. Take for instance, you've made sugar solution, right? Let's say you've made a sugar solution whereby you've put sugar particles into water. Now, let's say the concentration of sugar is higher than the water molecules. You've made what we call hypertonic solution. So now you realize that putting this cell into this solution, which is hypertonic solution means a solution whereby the solution itself is higher in terms of concentration than the solution inside of the cell. Okay, so when they say hypertonic, placing this cell into hypertonic, they simply mean to say putting this cell in a more concentrated solution than it is. So what exactly do you think? What is going to happen is that water is going to leave out of the cell, and after water has left, the cell will become what? Crenated. So crenation will take place. The effect is that this cell will be crenated. If at all we are talking about an animal cell, this is an animal cell. It will undergo what we call crenation. But if this cell was a plant cell, it would have undergone what we call plasmolysis. Sorry, plasmolysis. P-L-A-S-M-O-Y. Sorry, what am I saying? P-L-A-S-M-O-L-Y-S-I-S. So hypertonic solution brings crenation, cell crenation to animal cells. But to plant cells, it brings what we call plasmolysis. Yes. So we have the next question. This question is on saprophytic nutrition. Saprophytic nutrition. Okay. So the question says, figure 2 shows the structure of a fungus. And then we have the labels D, E, F, and G. D, E, F, and G. Then they're saying identify the parts labeled D and G. D is sporangium. D is what? Sporangium. E is sporangiophore. And I can tell you F and G. F is stolon. G is rhizoid or rhizoid. B, they are saying explain the functions of the parts labeled E and F. E simply puts the sporangium at the apex. All right. In short, it supports the sporangium. Sporangiophore supports the sporangia, the sporangium. Yes. How about F? F, which is a stolon, right? F is a stolon. So what exactly is the function of the stolon? You know, the stolon provides an aerostructure for the growth of the mycelium and the occupation of large areas. So this one simply provides an aerostructure for the growth of the mycelium and the occupation of large areas. Yes. Okay, C one, they are saying outline the process of nutrition carried out by the fungus in figure 2. Outline the process of nutrition. Outline the process of nutrition carried out by the fungus in figure 2. So this is the process. The process is that rhizopaths do what we call extracellular digestion to digest the dead and decaying matter. They secrete digestive enzymes into the surrounding environment and break down the organic matter into simpler substances. The nutrients thus produced are absorbed directly through the cell membranes of the organisms. Yes, rhizopaths, okay, rhizopaths undergo extracellular digestion to digest the dead and decaying matter. They secrete digestive enzymes into the surrounding environment and break down the organic matter into simpler substances. The nutrients thus produced are absorbed directly through the cell membranes of the organisms. And so we have the last question. The last question is saying, okay, where is the last question? The last question, give an example of a fungus that carries out the process of nutrition shown. Give an example of a fungus that carries out the process of nutrition shown. Okay, so this is the answer, rhizopaths. Yes.